All right, so we are getting into the special senses of the body now. Specifically, we'll get a lot of detail into the eye and ear, and then a little bit into smell and taste. We've talked a lot about the nervous system, receptors signaling neurons, communicating to different parts of the central nervous system, and the balance between peripheral and central nervous system. Let's get into the special senses, all right? So with the special senses, touch is a huge one. It's one of our big ones that we've talked a lot about, but we also are going to deal with vision, taste, smell, hearing, and then I've also included equilibrium here. Now, equilibrium is part of our ear. Our ear does two things. It does hearing and equilibrium, and we'll see how that works in a little bit. Where we're going to spend quite a bit of time first, though, is with vision. Right? So our eye. 70% of all sensory receptors that in the whole body, whole entire body, is in our eye. So our eyes are extreme, tons of receptors, sensory receptors, also known as photoreceptors, right? But when we're dealing with the eye, we're not dealing with just the receptors and how we see. There's a lot of other accessory structures for the eye. When we're dealing with the eye, we're going to deal with muscles with movement. We're going to deal with lacrimal glands, eyelids, and so forth. So this is a lot of lab stuff. You'll have to know all of this stuff for lab, dealing with our eyebrows, the muscles. I want to talk though about the conjunctiva. The conjunctiva is this thin layer that lines the eyeball itself as well as the inner part of the eyelid. The inner, and you can see this is all one continuous layer through here. This layer that is on the inside of the eyelids is palpebral conjunctiva versus the part on the eyeball, which is our bulbar conjunctiva. And when we're dealing with conjunctiva, the function of it is to produce more of a lubricating mucus through here and really helps with the drying out of our eyes. That is not just our lacrimal gland, that's our conjunctiva through here. When we get really red eyes, like for irritation, whether it's allergies, lack of sleep, that kind of stuff, those red eyes, the blood vessels, bloodshot eyes, that's the conjunctiva that has this vascular system in it. So that's actually what we're seeing in there, and it's really thin. This is also what can get inflamed in conjunctivitis, which is pink eye. Conjunctivitis, I guess is more proper way to say it. Conjunctivitis, which is pink eye. Yeah, so that's just different parts of the conjunctiva. Up in this lateral portion of the eye, you're going to come up with some lacrimal glands. These are tear glands, right? And if you notice, this is going to be on the upper lateral aspect of the eye, and it will drain across the eye and enter in these ducts through here. This is the lacrimal punctum and the caniculus that goes into our nose. So that's why when we cry, tears will actually drop down into our nose, and our nose gets a little runny from crying. Now, so this is a fluid formation and flow of lacrimal tears. And this is good because it contains antibodies, it contains like a mucus type fluid, lysosomes. This will help kill a lot of bacteria, foreign invaders, but it also helps keep the eye moist. It also takes debris and gets rid of the debris. Just a side note too is if you ever get something stuck in your eye, or a piece of something gets in your eye, not stuck in your eye, go to the emergency room, right? Don't remove it. But if you get like debris in your eye, you feel like an eyelash in your eye, you want to wipe lateral to medial, and that will help go with the flow of tears, and you'll be able to get it out of this punctum area for you. Yeah, so tears contain mucus, antibodies, lysozymes, drain into the nasolacrimal duct there. Now, we've talked a little about when we got into cranial nerves. There were 3 different cranial nerves that affected eye movement. We saw this chart. I asked you to start memorizing it already. Here we are again. Start really knowing what these are. A majority of them are cranial nerve 3 when we're dealing with the ocular motor, but we also have the trochlear and the abducens that will affect the eye as well. And they attach to the eye like so. Now, when you're looking at this, you can see 1 and then 2 on each side of the muscle. Like this one on the lateral aspect, this will be on the medial aspect of my eye. This is my nose. The medial muscle here is right next to that nasal cavity. And then you have 1 superior, 1 inferior. And then you're going to see 2 of them that come kind of across the eye like this. These are our rectus muscle, our oblique muscles that come diagonal. The rectus are these 4 that come directly off the top, bottom, and sides. Now, if you are looking at these muscles, this is better explained in lab. I wish I had a lab model, but instead, this is going to be my eyeball, right? All right, so this is my right eyeball. Now, on my right eyeball, if I have my lateral rectus and be on the outside through here, you can see is if it contracts, it will pull my eye and my eye will go laterally. Versus this my right eyeball, this is medial. If this contracts, it will pull the eye and look medial. Superior, if it contracts backwards, you can see it would cause the eyeball to look up. Same thing if it was on the bottom through here. So if it contracts, it pulls the eye downwards. So it's pretty straightforward. Lateral turns your eye lateral, medial turns your eye medial, superior, superior, inferior, inferior. Those are the rectus muscles. So lateral rectus, medial rectus, superior rectus, inferior rectus. Those are our primary 4, pretty straightforward on those muscles through here, okay? Now, let's look at that was lateral rectus, medial rectus, superior and inferior rectus. Let's look at these obliques because this gets a little tricky. Now, I also want to point out that the rectus, the superior and inferior, not just elevate, but it slightly turns it medially as well. So when you're dealing with this superior rectus sitting up through here, this is my right eyeball. It is slightly to the side through here. So not only is it pulling it back, it slightly lifts it up medially as well, because it's not just straight on the side, it's kind of at an angle like so. So it'll pull it back so the eye goes medially and superiorly. Same thing with the inferior rectus. The inferior rectus is going to pull it inferior and slightly medially on that, okay? Now, the inferior and superior obliques, those get a little bit more confusing because when we're dealing with this, they are opposite of what the rectus wants. The inferior oblique is going to elevate the eye. The superior oblique is going to depress the eye, and they both turn slightly laterally as well. So how does that work? Well, if you look at an eye, the rectus muscles come this way. This is my superior rectus on my right eyeball. Well, on the obliques, what happens is they come from the medial side and they come across through here and slightly anterior to posterior. So what will happen is when it contracts, the superior, it's going to pull the eye downward as well as lateral. So it's all about, and a lot of the pictures it shows it going straight across like so, but it's actually slightly pulling backwards. So the superior, it'll be coming in from this angle. So it'll pull it forward and to the side. Same thing with the inferior oblique. It is not just attaching to the side, it's slightly back through here. So when it pulls, it's going to elevate and go lateral. So those are our main muscles. No those actions. And let's get into the structure of the eyeball, okay? The eyeball itself is going to be, it's filled with fluids. We call them humors. There's three main layers. We call them tunics. There's a fibrous tunic, a vascular tunic, and sensory tunic. When we look on lab, you'll also see that broken down in the lab manual. And then you have a lens that kind of separates those cavities. Now, with our eye, we're gonna look at a few different structures here. Here is our eyeball. It's not completely round. You can see in the front, there is this little bulge through here of it. This is our cornea, right? And with our cornea, that turns into the sclera, and that is more of the white part of our eye, and then the cornea, the clear portion of the eye. But look through here. So that would be what we call the fibrous tunic. You can see this reddish area through here. That's our choroid. And that is going to be another layer, and then this inner yellow layer. Those are the three layers that we're going to be looking at and dissecting a little bit more. Okay, so let's look at this fibrous tunic first. The fibrous tunic contains what we call the sclera and the cornea. Now, this is not the full name, opaque sclera, clear cornea. This is just describing what color they are. So the sclera is more of a opaquish white, where the cornea is clear. We want to be able to get light through here. So this is our wider area, it's more posterior, and this goes all the way, wraps almost all the way around the eyeball, where our cornea is just on the anterior portion. You can see the cornea through here, all right? The cornea, let's see here. The cornea is, we call it like, it's the window. It's a clear window. And what we're gonna see is it helps bend light in different directions. So light bends when we shine it through things. If you look at through a glass and you have a straw, you'll see it kind of shift and the straw kind of looks weird angles. So light shifts and turns on different angles. Our cornea does that, but so does the lens. The lens is going to be sitting in deep inside the eye, and that will also help change angles of light. Where our sclera is the white portion of the eye, and you can see it wraps all the way around the back of the eye, even where the optic nerve comes out. And the sclera is where the muscles anchor into as well, those 6 muscles. Okay, so that is our fibrous tunic. Let's get into the vascular tunic. The vascular tunic, we also call it the choroid region, and there's a few different things in this vascular tunic. Choroid, we call it a choroid region sometimes, but there's other things in it besides the choroid. There's choroid, ciliary bodies, and the iris. So if we go back to this structure, here is that vascular layer. You can guess there is a lot of blood supply through here. It's vascular, right? And you'll see that this reddish color comes all the way around and deep inside through here. This is our ciliary bodies, and here is our iris. That's all part of kind of the same structure. Our iris is the colored portion of our eyes, what makes eyes green, blue, brown, right? Like that's that iris. Here, the ciliary body, you can see they almost have like little fingers here. And that ciliary body has these fingers, and then you'll see these white spiderwebby cords. These are actually ligaments, and we call them the suspensory ligament. And a ligament usually anchors bone to bone. Here, this ligament is going to attach to this ciliary body, which is has some vascular, and it's going to contract like a muscle right through here, and it also attaches to the lens. This lens moves. It's not completely solid, and it can get skinnier and wider and change angles depending on if the ciliary bodies contract and pull the lens up, or relax and let the lens drop down. So there is your 3 different portions of that. Um, in the ciliary, we call it the ciliary body, there is ciliary muscles in here, as well as the ciliary processes, which are the finger-like extensions off the muscles that the suspensory ligaments attach to. Now, let's go back to this iris. This iris here drops down, but can you see in this region, this is our pupil. See, our pupil, there's nothing there. We can actually see the iris would wrap in a complete circle, and really what the pupil is it's an empty spot. It is an empty spot, and it looks black because we're seeing into a very dark chamber. And when a doctor looks in your eye, they're shining a light with a magnifying glass to look through this hole so they can actually shine light on the back of the groove here and see what's there. So the pupil is actually just a hole, right? And this will contract or dilate depending on how much light we want to enter our eye. When it's constricted, it allows a little amount of light. When it's dilated and big, it'll allow a lot more light. And that's all about focusing light on the back of the eyeball so we can see. For that, let's see here. Coming back there, smooth muscles, sensory. Okay, how much light enters in there? Close vision, bright light, the pupils constrict. Distant vision, dim light, the pupils dilate. So a lot of people recognize, okay, yeah, bright light, my pupils are small, dim light, my pupils are bigger. But how close you're looking at something and how far will also affect the light, because that will also affect how much we're able to see and focus light on the back of the eyeball. Now, we also put here changes in emotional state can change your pupil dilation and constriction. So fear will cause the pupils to dilate. Sometimes when we're really attracted to something, they'll dilate, or when we're thinking. So different emotional states will affect constriction and or dilation and contraction of the pupils. Big part of that is our sympathetic and parasympathetic. When we are in a flight-fight mode, we want to be able to get as much light in there so we can get as much vision as we can. It's not as clear though. So we can see more in our peripheral vision with it wide open, but it's not as clear. Versus parasympathetic, we can contract, and then that light gets focused a little bit more on the sensory areas that deal with acute vision, and it'll be much more clearer, but we won't be able to see as much in our peripheral vision, versus our sympathetic, not as clear, a lot in our peripheral vision. Okay, so that is our pupils and more of our choroid region. Now let's talk about the most of the choroid region. That was the ciliary bodies, the iris, but look at all of this. This is a giant portion of the choroid vascular layer. The choroid is filled with blood. It's a darker, this looks red, but it's more like a darker brown. Its function, let's go through here. Its functions. Its function is to provide blood and nutrients to the eye, big one. But this darker color also helps absorb light and helps it when light comes into the eye, it's going to hit some of the sensory, it'll get through some of these, and this area helps absorb the light so we don't get reflection. Can you imagine if this kind of reflected and didn't absorb light and acted more like a mirror? You'd get light bouncing all around and we wouldn't be able to focus the light as much. So this helps absorb light and prevents scattering of light throughout the eye. So nutrition and preventing of scattering by absorbing light. Those are the two main functions of the choroid region itself. So that is our fibrous, our vascular. We're going to get into the sensory in a moment, but the other thing I want to point out here is this internal layer, or this internal cavities are filled with fluids, And the lens is what separates the internal cavities from anterior and posterior segments. So you can see here, why this shows like strings, this is pretty solid. You're not getting fluid to pass through here. So in front of the lens, this whole area is filled with a type of fluid. We call it an aqueous humor. It's much more watery-like, versus behind the suspensory ligament in the lens, all of this is what we call vitreous humor. And this is a thicker fluid. And this is gonna help with focusing light as well. And you can see how the lens separates the anterior segment and the posterior segment. And let's get into this sensory portion, because this is really a huge part of what we want to talk about, okay? So the sensory tunic. The sensory tunic is the retina. A lot of us have heard about the retina before. The retina is what we call the sensory region of our eyeball, okay? And that is going to look like this yellow portion through here. These yellow portions, you can see, also travels in through here, and that's our optic nerve. All these receptors are going to receive light and information and send that information through this optic nerve, and that optic nerve will then go to various parts of our brain and eventually end up in our occipital visual functional area to interpret what we're seeing, okay? But in this retina, there is multiple different layers, and this is what they're going to look like. We have two layers. One is the pigment layer. This is really thin, and then the neural layer. Pigment layer, very thin. The reddish colors are choroid. The majority of these cells that you see here are neural layer, and then a slender area is our pigment layer. I'm going to take this a little bit closer. So here are the different cells that are involved in the neural layer. And then this portion through here where we see these little cells within nuclei and some dots, those are our pigmented area. Now, what is in our neural area? Well, I just showed you three different cells. We have photoreceptors. Those are the cells, receptors that are receiving light and turning it into energy. And then those connect to what we call bipolar cells. We've seen bipolar cells when we talked about the different types of cells, unipolar, bipolar, and multipolar. Well, here are your bipolar cells, one type. And then that turns and communicates to the ganglion cells. The ganglion cells have the long axons that carry that information to the optic nerve. So let's take a look at that, okay? Here we go. With our cells through here, I'm sorry. This is the inside of our eyeball. So this is where it gets a little confusing. Here's the inside of our eyeball. We'll take it in different layers. Here's the eyeball. We're going to take a little bit closer peek here, and then even closer here. I'm sorry, here. So light is coming in from this direction, right? We're going to be coming in from here. That's what we're seeing on this arrow, right? Light's coming in. And what is strange, and it's kind of a hard concept to get, we typically think about, okay, it's immediately hitting the cells, which will communicate to another cell and then to another cell, but it doesn't, right? Instead, light goes all the way through all of this, and these cells through here are picking up the light. They're going to turn that light into an energy and then go back out this direction. So these cells, these photoreceptors, then communicate to the bipolar cells, which then communicate to ganglion cells. Those ganglion cells turn that into an action potential down their long axons, and those axons end up lining the inside of the eye and traveling out the optic nerve. So let's come back, clear that out. So you'll see light comes in to that pigment and then it goes back out, bipolar ganglion, and these are all different nerves that eventually axons then turn into that optic nerve. All right, so that is the direction of light. Now, let's look at that a little bit closer. Now, on those photoreceptors, we have two different types of photoreceptors. We have rods and cones. Rods are going to be focusing on more of our what do we put? They're sensitive to all light. They respond to all light. They're more sensitive to light. They're also much more numerous. They're used really good for in dim light, but it's also our peripheral vision. Since they're all over the back of our eye, really sensitive, we're going to be more sensitive to everything we see. Cones, however, their response is to bright light. That's why when we are in dark lighting, things aren't really clear. It's because we're using these rods. In bright light, it's much clearer, and this is where we're getting color from. Rods are black and white. We're seeing more of we don't see the in grays, that gray spectrum. Cones is where we're actually getting our light from. And they are going to be seen in three different types of colors. So in these cones, they're located just in the back of the eye, where rods are located everywhere. And these are going to get centralized. And the more it gets centralized in the very back of the eye, we call that the macula lutea. The macula lutea is this smaller portion on the back of the eye. Let's see if it's through here. Yeah. So it's directly behind the cornea and the lens. If you go straight back, that's where your macula lutea is. And that is going to be loaded with cones. Some rods, but definitely a lot of cones. All of this other stuff, almost all rods. Now, in the very center of this macula lutea is your fovea centralis. Your fovea centralis is the highest concentration of pure cones. Lots of vision for color and preciseness and sharpness. And you can see that light, when focused, it'll go straight back and hit this fovea centralis. This optic nerve is not directly back. It's off to the side a little bit. Okay, so this is where we're seeing acute vision. Now, when we are getting into these rods and cones, the cones are going to be coming in three different colors: red, green, and blue. That's all we have. Those are like the primary colors, right? And there's a combination of those colors that all other colors arise from. Anyone who's taking color theory classes, which I have not, but my wife did when she was at the Fashion Institute, right? Like all the different spectrum of colors all comes from red, green, and blue. And therefore we have those cones. And people that have different color blindness, it's because they lack a certain color of cones or a certain number of that specific color. They may have a lot less red cones, and so it's going to affect their color vision for that. Okay. How do we see other colors? It's all about what cones pick up. So if we saw a lot of yellow light, we're going to get both red and green cones being stimulated, which tells us that that's yellow light. Now, if the red cones are stimulated more than the green, then we're going to see more of like an orange color. So it's all about what cones are being stimulated to absorb that energy and pass the information on. Now, here's what I want to point out. If you look, this is just showing a cone through here, right? This is a cone. Yeah, we're making it a yellow cone, and there is no yellow cone, right? It could be either red, green, blue. We're not really differentiating what kind. We're just calling it a cone. Look how one of these cones, one cone attaches to one bipolar cell, which is going to communicate that to the ganglion cells. You can see here on these rods, there's multiple rods that will attach to one bipolar cell to a ganglion cell. It shows like 2 rods, 3 rods. There's a lot more on this. We can have hundreds of rods connecting to one bipolar cell. So that is a lot of photoreceptors going into one cell, into a ganglion cell, to stimulate an action potential. This is also why we see with rods aren't as acute. They're not as focused. There's not as much, there's a lot of information going into one cell versus a cone is going to have one cell into one cell. And that will really help with that precise, clear vision and color that we're seeing. And that is our sensory. Now, when we have all these ganglion cells, the axons come in through here and travel out this optic nerve. Well, look here, there is no photoreceptors where that optic nerve comes out. There can't be, because that nerve has to go out the eyeball. This is what we call the optic disc. There is a small circle in the back of the eye where the optic nerve comes out. And let's see if I have a picture of that. I don't have a picture of it, but if you look in the back of an eye, you're gonna see a small circle, and that will be the optic disc. This is also called the blind spot. So there is a part in the eye that light, when it hits, we do not see anything. You can see that, right? So if light's hitting that area. Here's the cool thing is as we're looking at something, we do not see this black little spot that is considered our blind spot. There will be an activity for a lab experiment that I want you to do in your lab book that has you look at something on a page, and you'll get it, you bring it forward and backwards, and all of a sudden that spot that you're looking at, it's a little plus, will disappear. It'll just disappear. And so what that shows is that is where you're concentrating that light from that little X that you're looking at. You get it in the specific spot that light centers on that optic disc, and you can't see anything. But the reason why we don't see that black spot all the time is because our brain fills that in. It just blurs it in because we see all the surrounding areas, and our brain makes up that difference, which is pretty stinking cool when we're looking at it. So now let's get into the lens. So we've now have the back of our eyes. We looked at the few layers of how we're receiving the light, what's being stimulated that passes, turns into energy, turns into action potentials, travels to our brain to process different things. It doesn't, just so you know, we'll get into a little bit in a little bit, but that nerve doesn't just go directly to the occipital visual functional area. It goes to the thalamus first for processing and then gets stimulated, but we'll look at that in a moment. What I want to do is, now that we have light, how do we focus on the fovea centralis and the macula lutea? Like, how do we get it into that? We call it the macula most of the time, like that macula and fovea centralis. How do we change the shape and angles of those light and not just have it scattered all around? That is the function of our cornea, but especially the function of our lens, okay? So when we are looking at light, let's just look at light. If I had, let's look here. If I had a concave surface versus a convex surface, if I shine light through here, what will happen is you can see light would come here and change its angle and slightly move in a different direction, versus a convex would come here and then change the angle. That's all about different angle changes that we see through here. Okay, so concave, convex. The lens is a biconcave structure, meaning it's rounded on both ends. Biconvex. I'm not sure if I said concave. Biconvex. So it's more of an oval shape. It's transparent. It is flexible though, right? It does move, but it doesn't have a vascular supply. This is going to make the precise focusing on the light so we can get it on the back of the retina, in the macula, and the fovea centralis. However, this flexibility becomes a little bit less with age. We lose that elasticity, and that's often why we see vision go as we get older quite a bit. So let's, and then when they really become less movable and don't get the vascular supply, we can actually have that clear transparency start to become opaque and hard, and that's what we call cataracts. So here would be a cataract. You should be able to see through here because here's the iris, the pupil shining right through here. The lens is behind all this, but we're seeing through it, versus here, and here, we can't see that dark area because the lens is right there and it's this opaque thing. So that's cataracts with a light. Now let's look at this lens. The way it changes shape is what is a process we call accommodation. And that is going to help us focus on things up close and far away. And what happens is when we see something up close, the ciliary muscles of the choroid vascular layer, right? The ciliary muscles of the choroid body are going to contract. And if they contract, it makes this lens a little bit rounder. When it's relaxed, it pulls and it gets a little bit elongated. So here's this lens, and if we contract on it, it's going to pull it, it's going to pull it down and make it a little bit more rounded. And then when we relax, it pulls, it allows it to go back to its natural shape, pulls up and elongates it, okay? So that's what we're seeing on that lens. So let's take a little bit closer peek at how that would affect. And let's see if I can draw something. So let's take an eyeball like so, right? And then we're gonna put, let's put a lens in there. In its relaxed state, a lens is nice and long and skinny like so. And so if we shined light in, it's going to change a little shape with the cornea. It would go in, but I'm just going to show it go straight in like so. All right, so here's some light coming in, and we'll hit it from a couple different angles. Now, if we have light coming in and hitting that lens, if we take it, it's going to change the angle. It will. So if we change the angle, it'll change it a little bit at an angle and drop it back. Same through here, it's not going straight, it slightly comes in at an angle and drops back. Now, what happens if we change the structure of that lens? So let's take the same eyeball. So we're going to take that same look of that eyeball, make this a little bit smaller so we can see it side by side. All right, I don't know. Can't get it all the way through there. So I wanted to get it all on the white background. Let's try one more thing. Oh well. So we have this pupil through here, shining light, and then our lens. Well, what happens when we change the lens to a thicker lens? So now we're contracting the ciliary muscles. You have these light rays coming in, just like we did before, right? But now all of a sudden, we have this lens that is contracted, and these angles change completely. So rather than going through, it would contract, they actually curve even more. And now all of a sudden, our light is not reaching the back of the eyeball. So we can change the angle depending on what we're looking at. So far and close things, we'll be able to, if something's close up, we'll be able to contract it, and then it will make these go towards the back of the eye where we typically wouldn't see stuff. You do not have to understand what happens on all of these. I just want to give you an example of how the lens changes the shape of the, how the lens changes the angle of lights. There we go. Whew, finally. So we can see how this works, right? So I wouldn't have you write down, have you draw something like that, but just know if I change the shape of the lens, the angle of the light changes with that. Okay, that's the whole purpose of the lens. However, what I do want you to know is this, where if it contracts, the ciliary muscle contracts, the lens rounds, and that's good for close-up vision. Versus distant vision, the ciliary muscle relaxes and the lens flattens. I do want you to know that, okay? All right, so let's take this a little bit further now. So now we have this area of the two different lenses. Well, what happens here? Let me get rid of this. Let's go closer up. Now, what happens if we change the shape of the eyeball? So this is a nice oblonged eyeball. Well, what happens if I had an eyeball shorter like this? Well, the lens would be focusing on light that would actually go past where the fovea centralis and macula is. Well, what happens if I have an elongated eye? Well, the light is now focused too short of that. This is what we call short distance, or farsightedness, and nearsightedness. It all is about the shape of our eyeball. So when we are looking at, let's get back here to our keynote. So as we're looking at our eyeball, here are the different shapes of the eyeball. When we have something that's called short or nearsightedness, it's myopia, and the light is shining, and because of the shape of the lens and the eyeball, it stops a little short, and we can see things better close up. Where farsightedness, the shape of the eyeball and the lens makes the light shine beyond that, and that's when we would see something a little bit further away. So nearsightedness, farsightedness. And then you can see lens-wise how they would correct that. They would correct it through more of a convex lens for nearsightedness to help bend the light a certain way, where that's farsightedness. Where nearsightedness, they do more of a concave lighting lens. So you can change the lens to change the shape of that. Yeah, so that's how that works. Really, though, understand that we're having all kinds of changing of light because we have it coming through the cornea, which changes the angle a little bit. You're going to see it change through the aqueous humor a little bit. It hits the lens, it changes angle. It hits this vitreous humor, which changes the angle. There's various different things that are going to change this light. But our lens is the one that we control by the contracting and relaxation of the ciliary muscle here. So let's look at this whole pathway of light. So if I said light was going to come in, what would be the whole pathway? Let's go through that whole pathway. So we have light comes in, hits the cornea. The light then goes through the cornea to this anterior segment filled with aqueous humor, goes through the pupil to the lens. Through the lens, it goes through this into this posterior chamber, which is filled with vitreous humor. Then it hits the back of this eyeball, and it goes through, let's go up here a little bit closer, it goes and passes the ganglion cells, the bipolar cells, gets absorbed into these photoreceptors. Those photoreceptors pick up that light, convert it into energy, go back to the bipolar cells, to the ganglion cells, to the axons of the ganglion cells, which travel through the optic nerve and out the eyeball. Be able to state that whole process of how light travels through the eye, right? Now, also recognize as light traveled through and got picked up by these photoreceptors, there's some that gets past that, and that's where this pigment area helps with some of that scattering, as well as the choroid vascular layer provides some also absorbing of the light. All right, so now that we have this light that is converted to action potentials and information going down the optic nerve, where does it go from there? Now, here's what happens. The axons of the ganglion cells form that optic nerve, and then that optic nerve is going to go into our brain, and it has this chiasm. So if you remember from the portion that we are dealing with the central nervous system, maybe even in lab, you're going to see that optic chiasm. That optic chiasm is the place where the nerves can cross, okay? So we're going to have vision come in, and some of it will cross to the opposite side. Some will come in and branch off to the same side, depending on what part of the eye it's hitting. So let's look at that a little bit more. Here's your optic chiasm. Here I'm going to show you how some of it crosses to the opposite side of the brain, some stays on the same side. We'll get to that in a second. But where does all that information go to? Well, most of the fibers are going to go to that thalamus, what I talked about. This thalamus is this relay center. It's the relay center for almost all sensory. It gets processed there, and then it will get sent to the visual cortex, which is in the occipital area of the brain. However, some of these optic tracts also go to the superior colliculi. The superior colliculi is in that midbrain area, and that's going to help with our biorhythms as well. So helping with the biorhythms, natural states of light and darkness. Okay, so let's look at this. How does it get to the thalamus for this and the superior colliculi? But again, most of it to the thalamus. Well, here we have visual pathways. If we are looking, this would be our right eye, this is our left eye, you can see the medial part of our eyeballs go to the opposite sides of the brain. The medial aspect of an eyeball is absorbing light from more of the lateral aspects. So if we're seeing laterally, that light shines in and hits the medial part of our eye. That medial part gets transferred to the opposite side of the brain, versus more of medial light will hit the lateral part of our eye, and that information goes to the same side of the brain. So here we are looking more on this this area. So this would be the more left side of the visual area that we see, this left side. Well, this left eye is going to pick up that information from its lateral aspect and bring it to the right side of the brain, where this right eye, the left visual area, is picking up, and it's picking it up on the lateral aspect and sends it to this right side of the brain. Can you see that? The reason for this is this helps us with three-dimensional and depth perception. So it helps us with depth perception, see things in more of a three-dimensional pathway is how this would work. And you can see here is that superior colliculi. We have them, and that some fibers will branch off and go to the superior colliculi, but the most of them are going to be going to this thalamus area, which is here and here. And then if you look closely here, you're going to see those nerves end and then communicate to new nerves that bring and process that information to the occipital lobe, the primary visual cortex. And that is our eye.